So Dr. Chris Costello, who's head of the laboratory compliance at Medimmune, giving us a pharmaceutical industry interview.
Thank you very much. Is that about right?
It's okay.
Good morning, everybody. It's a sad reality that there is a lot of disease in the world. All of us in this room, in one way or another, are involved in getting medicines to the marketplace to treat patients. Be it like we do in the pharmaceutical industry, we discover drugs, develop them, get them to the market. But the general research community, they study biological functions to understand disease. So biological samples are an essential reagent for developing drugs, getting them to people, making patients better. I'd like to thank Christine and the organizers for inviting me to give a talk today. It's a real pleasure to be with this group because it's the first time that I've attended the Stratified Medicines group, so I'm learning a lot. I have a few slides that I want to share with you. I hope that I'm not going to end up teaching my grandmother to suck eggs, but there are some simple concepts that I will get across to you. It's almost lunchtime, so those concepts are relatively straightforward. You don't have to think, and we'll hopefully get to lunch relatively quickly. So this is the outline of my talk. I'd like to give you a background on Medimmune. I think it's important for everybody to understand a little bit about Medimmune and a little bit about the pharmaceutical industry, what it is that we do, why biological samples are important. And then I'll go on and say why biological samples are important to us and why biobanking is important to the pharmaceutical world, just as it is important to the research world. Okay, here we go. So quick overview of Medimmune. Medimmune is a member of the AstraZeneca group. We form what is known as the biologics arm of AstraZeneca. So we make large molecule medicines, predominantly antibodies. They're big biological reagents. We have quite a robust pipeline, more than 120 biologics in research and development at the moment. We have 30 projects. Our key areas are respiratory oncology, but we do have interest in metabolic diseases, vaccines and neuroscience. So quite a broad spectrum of disease focus. This is a timeline, just to give you a little bit of a potted history of Medimmune. It's been in existence for a while. I don't want to sort of go through the timeline. I just want to pick out some highlights, if you like. If we go back to here, in 1993, as it was, Cambridge antibody technology was in a collaboration to work on TNF-alpha receptor and antibodies to TNF-alpha receptor. That led to a drug called Humira, which is now generally used across the world. It's a leader in arthritis. It's actually a multi-billion-pound drug. Another highlight, just to show you, is in the vaccine front, there's a flu, something called flu mist, which is a seasonal flu vaccine. A new one comes out every year. The difference between this vaccine is that it's actually intranasally administered. So you don't have to have injection, you actually sniff it up your nose. Just to go on and talk a little bit more about the therapeutic areas that Medimmune is involved in. I mentioned already the three key areas are cardiometabolism, oncology, respiratory inflammation, and autoimmunity. But we do have interest in infection and neuroscience. But those are more sort of opportunity driven. We look for areas where antibodies can offer something that other modalities do not offer. And we go across the board, biologics, small molecules, immunotherapies, and protein engineering. This slide is really just to indicate the depth of the pipeline again, in oncology, respiratory, CVMD, infectious and in neurosciences and in infectious diseases. So we have compounds across the board, if you like. Lots of in phase 1, phase 2, some in phase 3. So we hope in the next 2-3 years, some new drugs will be coming on the market in both in cancer and respiratory. And most recently, the quadrivalent flu vaccine was approved. Okay, so let's change tack. I just want to talk a little bit about biological samples at Medimmune. The titles here, don't get too hung about them. Really, what they refer to are the areas where we store the samples. But I'll tell you a little bit about each area. So in Cambridge Pathology, we have what we call the Human Tissue Bank, where we store essentially frozen tissues or paraffin-embedded tissues, tissue microarrays, and so forth. And they predominantly used for immunohistochemistry. Then in the next box, the yellow box, RMS Human Sample Application, that just stands for the database that we store the samples. RMS stands for Research Management System. But here, the samples are essentially blood-derived samples. They tend to be liquids. They are blood cones from the National NHS Blood Transfusion Service. We have an internal blood donor panel, and we take blood when we need fresh blood, for example, for neutrophil assays and so forth. And then the last box there is our bioanalytical group, where we predominantly store samples that are serum or plasma, which come from clinical trials, and they're generally used for pharmacogenetic studies or metabolism. So those are the major reasons that we have, or the major areas that we store samples. But those samples are used across the board in the drug development process. So they're used in identifying potential new drugs. What we do in Medimmune is predominantly something called phage display, where we identify antibodies by challenging a library with the target. And so we can use cells to do that. So human cells are important in that part of it. But they're used in screening assays after a lead is found. I mentioned immunohistochemistry. We can use predominantly cell lines at the moment to build mouse models. But going forward, we're looking to use actual human derived samples, fresh samples for that. And a critical reagent for us is the fresh blood, because some of the assays that we need to develop require, for example, neutrophils that are very short-lived. And so we need to have an in-house blood donor panel where the sample is collected and used within sort of 30 minutes, so less than an hour usually. So that's like a potted history of where we are with Medimmune. I want to spend a couple of minutes now to talk about the STRATUM Project. This was and I'm going to use the word initiative because I know Alex didn't like that word, but I want to say that yes, there have been lots of projects in this area, and one of those projects is the STRATUM Project. And why did that come about? Well, simply as the boxes say, there are insufficiently well characterized human biological samples, or perhaps the visibility of sufficiently well characterized human biological samples. And there is a complexity in biobanking. I hope you will all agree with that. So the STRATUM Project was born out of that need. There was funding from the Technology Strategy Board, as it was then. It included private and public input into the project. There was, for example, AstraZeneca, GlaxoSmithKline. We had Manchester University, we had Leicester University, we had Nottingham University, to name a few. But the goals there were to try and resolve this problem, this issue of insufficiently well-characterized samples, excessive complexity. There were some outputs. There were several work streams there. There were some outputs around best practice, some framework for governance, and a cost model that Martin will talk about this afternoon. There were some standards and guidelines, ideas around bioinformatics and catalog and so forth. Now I'll say that, and I'll say in the same sentence, the Confederation of Cancer Biobank did some fantastic work along the same lines, and there is a standard now available for biobanks to try to bring some uniformity and some harmonization in how biobanking is done. I don't know where we are. Is that the answer? Perhaps the next slide can help. So in this slide, I've tried to sort of bring together what the pharmaceutical companies would like, what Medimmune, AstraZeneca would like from biobanking. I'll probably lead you to this here, which is a CRC UK Tissue Directory and Coordination Centre. I was delighted to go to the launch of the directory 3-4 weeks ago in the Royal Free Hospital. Fantastic to see that perhaps all the strands are being pulled together, they're all coming together. It would be lovely to imagine that. I think it's still a complex area, there's still lots to do, but the question is, do we buy into it? Do we want to succeed? Do we want to have a uniformity? Do we want to use standards? Do we want to work together? I'll say, there are lots of standards out there. The STRATUM Project provided standards, the CCB provided standards, European organizations have provided standards. There's lots of stuff out there. It is still very complex. How do we simplify? That is my question to you. Actually, while I'm down on the bioethics, I haven't got consent on there. So I'm going off piece now. It wasn't planned to say this, but we heard from Jane how difficult it is to get that informed consent. Maggie said that we mustn't forget that samples come from real people. They are human beings just like you and me. But I would challenge this group. Why is it so difficult to get this informed consent? I mean, STRATUM Project provided templates of various colors. CCB have worked extensively on informed consent. Other organizations have. Is it really an intractable problem? Maybe this is something for us to discuss in the afternoon, but to me, standing back and watching this today, sounds as though it's a very, very difficult problem, but is it? Are we making it more complex than it really needs to be, is my question to you. Really, that's all I wanted to say, other than to sort of give you food for thought, perhaps bring up some thoughts for the discussion this afternoon, and just to remember, human biological samples are essential elements of research. They're absolutely critical to developing medicines. There isn't a question about that. So thanks for listening. I'm happy to answer any questions. Thank you.